Hello students! In our today's video, we will learn about the process of pollination and fertilization. In our previous video, we learned about the flower as the reproductive organ of the plant. It consists of an androecium or stamen and gynoecium or pistil. Each one of them is made up of reproductive structures like filament and anther, stigma, style and ovary respectively. Now the stamen has an anther which bears pollen grains, the male gametes, while the pistil has an ovule, the female gametes. The pollen grains from anthers are transferred to the stigma of pistil or carpel, and this process is called pollination. Thus, transfer of pollen grains from the anther of a stamen of flower to the stigma of a carpel on a flower is called as pollination. After pollination, the next step in the reproductive process is the fertilization of this pollen grain with an ovule in plant's ovary, which leads to the formation of seed and fruit. So let's see this fertilization process in plants. After the pollen lands on a suitable stigma by pollination, it has to reach the female germ cells that you see here inside the ovary. Now this is the pollen grain which reaches to this female germ cell through the style of the carpel. For this, it begins to germinate and the tube grows out of the pollen grain and travels through the style to reach the ovary. The small tube-like structure is called the pollen tube. It emerges through the germ pore present on the pollen grain. The contents of pollen grain also move into this pollen tube as it grows towards ovary. The pollen tube grows through the stigma and style and finally reaches the ovule through the micropyle region. Yes, student, the gap that you see here is micropyle, and through this gap, pollen tube enters inside the ovule. At this stage, the vegetative cell in the tube degenerates, and the generative cell divides to form two male gametes. After this division, the tip of the pollen tube bursts, and two male germ cells that were present enters this embryo sac. One cell fuses with egg cell and forms a diploid zygote. This fusion is called a syngamy. Remember students, the fusion of pollen grain with egg cell from the egg apparatus is called as syngamy. The other cell fuses with the secondary nucleus which is present in the center to form the primary endosperm nucleus. Now observe here that this structure is triploid. That is, it has three sets of chromosomes represented as 3N. One from male germ cell and two which were already present in the nuclei. And hence it is called as triple fusion. Note here that primary endosperm nuclei does not degenerate but develops into endosperm fluid, which is the nutritive fluid for the developing zygote into embryo. Since two types of fusion or fertilization, that is syngamy and triple fusion, takes place within an embryo sac, the process here is termed as double fertilization. Double fertilization stimulates the ovary to develop into fruit via seed formation. It also helps in the development of new characters from two different individual plant species. Now students, after this fertilization, the ovary undergoes certain changes. So let's see them. After syngamy, zygote is formed and continues to develop as seed. Thus, the ovule develops into the seed. This seed grows into the new plant whenever it is allowed to germinate. The integuments of the ovule develop into the seed coat. Observe that this is the seed and the layer present outside it is called as seed coat. It is tough and protective for the seed. It protects the seed from harsh conditions within the soil. Thus, the seed contains the future plant or embryo, which develops into a seedling under appropriate conditions. As the zygote develops to seed, the primary endosperm nuclei also forms endosperm fluid. After triple fusion, the primary endosperm nucleus develops into an endosperm. The endosperm provides food for the developing embryo, and the whole ovary enlarges and develops into a fruit. Later, the remaining cells called the synergids and antipodal cells degenerate. Antipodals and synergids degenerate only after the double fertilization. But this does not mean that they are functionless. Well, antipodals guide the developmental process and the synergids perform two functions. One, they provide nourishment for the movement of generative cell towards the polar nuclei. And second, they guide the entry of generative cell within the embryo sac. Now students, observe the image on your screen. It is of watermelon and the other is of maize. Can you spot seed, endosperm and fruit present here? Pause the video, take a second and give it a shot. Could you? I'm sure you must have done it. So in this video, we have seen how sexual reproduction in plants happen. The first step of reproduction is pollination, which is when pollen grains sit or fall on stigma. And the next step is fertilization, which is when the male germ cell unites with the female germ cell or egg cell inside the ovary of a plant. After this, zygote is formed and fruit and seeds are developed from the zygote. And this whole process is sexual reproduction since male and female gametes are involved. In the next video, students, we will learn about sexual reproduction in humans.